Hey guys, hope everyone's doing well this morning. Thanks to Edward for inviting me and for y'all having me out. I know y'all have been sitting here listening to other people talk all morning, so bear with me for just a little bit longer, and then it'll be lunchtime, I guess. So like they said, my name's Kyle Montgomery. I'm a consultant and agronomist for Advancing Eco-Agriculture. I live up in Surrey County in the northwest corner of the state, where it was a balmy 10 degrees this morning when I left. But I work all throughout the southeast and am starting to work with some more folks here down east. So for those of you who don't know about AEA and what we do, I'll give you a bit of an introduction real quick before we get into soybean nutrient management. We were started by a fellow named John Kempf back in 2006 up in Ohio. John's got a great podcast called the Regenerative Agriculture Podcast. Some of you may have heard of that, and if you haven't, I encourage you to listen to it. There's a lot of really good information and interviews with growers, and they talk a lot about the future of agriculture and just all kinds of good things. So check that out if you're into podcasts. But AEA has been a pioneer in regenerative ag for really since 2006, so longer than a lot of other companies out there. And our work, the scope of our work really is we're a nutritional products and consulting company, and we partner with growers to help them implement practices on their operations that help them become more sustainable, grow better crops, reduce inputs, and ultimately become more profitable. We've worked on millions of acres and nearly every crop out there, and the big sort of different angle that we take is that we found that the root cause of the biggest issues that most farmers are facing and dealing with, whether it's insect pressure, disease pressure, or crop quality issues of any sort, the root cause of that can really be found in the lack of the loss of robust soil biology in our agricultural landscapes and the suboptimal nutrition of crops throughout the season. So our real focus is addressing these issues through precise nutritional management and using precise testing technology that's industry leading and state of the art, and then applying high quality bioavailable mineral nutrition at the right time, giving the plants what they need when they need it. Sort of philosophically, we believe that plants can be completely resistant to insects and diseases, and collectively we really don't know what a healthy crop looks like anymore, because in general crops are not, in our opinion, as nearly as healthy as they could be. So this really brings me to what I want to talk about today, which is how our recommendations for doing something about that and our approach for doing that. So the first thing, I want to explain some of the agronomic principles that we use to think about the nutritional requirements of soybeans, and really some of this is going to apply to any other crops that you grow, whether it's corn, other broadacre crops, or what have you. The second thing, I'll talk a bit about what our standard nutrient program looks like and the expectations and benefits and the thoughts behind that. And then third, I'll talk a bit about what I've seen in the field this year working with soybeans specifically here in North Carolina and some other growers throughout the region. So kind of like I said, as a bit of a preface, I guess, before I get into some of the agronomy, we put a large emphasis on precision nutrient management, and the reason for this really is that crops require, of course, and this is sort of obvious, different nutrients in higher volumes at different points in the growing season. And we found in many cases that a lot of the biggest issues that growers are facing come from the excessive application of a specific nutrient or product at the wrong time. And so we want to sort of shore up the timing of applications and help y'all as growers understand the effects and the broader implications of what we're putting out there on the greater landscape that we're dealing with. An example of this is we found that often large amounts of nitrogen or potassium put out pre-planting has, in some cases, very negative impacts and causes antagonism with other nutrients that are really needed in higher quantity, like calcium, early in the season. And I'll get into some of the details, specifics of that in a little bit. So we also like to say at AEA that we like to test rather than guess. So shooting in the dark and sort of shooting from our hip is not really how we like to do things, and we use and employ some very precise testing technology like plant sap analysis, which I'll go into in a bit, to really understand the nutrition of our crops in season so we can make informed management decisions. The last kind of bit I want to mention is we take what I think of as kind of a plant-centric approach to crop nutrition. There's a lot of talk about getting the health of your soil right in order to produce a healthy crop. And while that's very true, we can balance our minerals, we can get carbon in our soils, we can cover crop, we can increase soil biology, add humates, all these different things. At the end of the day, your goal and our goal is to grow a healthy crop. And you can't have a healthy soil without growing a healthy photosynthesizing crop on it. Because the plant is kind of the mechanism of bringing new energy into the whole agricultural system. It's the sort of solar panel that absorbs solar energy, sunlight, transforms that into carbohydrates, and sends some of that down into the root system, bringing energy, feeding biology to the soil. And so it's just a bit, it's a situation to me where you can't have healthy soil without a healthy crop, and you can't have a healthy crop without healthy soil. So there's a dynamic there, there's an interaction, and that's threaded through our philosophy and what we do at AEA. So we'll start off with photosynthesis. I mentioned that. There are kind of three processes in plants that we use that kind of inform our decisions as we think about nutritional management. And those are photosynthesis, protein synthesis, and grain formation or grain quality issues or fruit quality, whatever the harvest product is. In this case, of course, it's grain. Photosynthesis, obviously, is what I just said, the process of plants turning sunlight into energy. And that energy is in the form of sugar, carbohydrates, and they use this energy to do all the things that they do, to put on biomass, to produce leaves, to produce flowers, to produce grain, to fill grain, to send up to 25% down into the root system to feed the soil biology, to feed to invest in the health of the soil. But we often make the mistake, I think, of assuming that just because a plant is actively growing, just because it's greening up as a response to an input or just part of the growth of it, that photosynthesis is happening as optimally as it can. And it's our belief, and studies have been done to show that really, in most cases, plants are photosynthesizing, producing their energy at about 15 to 20% of their total capacity. So what would it look like if we could increase that amount, increase the energy in the plant, increase the efficiency of photosynthesis up 30, 40, 50, 60%. So in each 24-hour photo period, each day that a crop is out there under the sun, it has a chance to absorb 100% of the sunlight that's hitting it. But it's usually a quarter of that, or less than a quarter of that. And so what kind of improvements could we see if we increase photosynthesis? There are 5 primary required nutrients for photosynthesis to completely occur, and that's nitrogen, phosphorus, magnesium, iron, and manganese. And each one of these plays a critical role in the absorption of sunlight, the formation of chlorophyll, and then the cascade of reactions that lead to the formation of sugar. The reason I'm saying all this really is if on any given day, in any given 24-hour period of time when the plant is absorbing sunlight, one of these nutrients isn't in as abundant of supply, isn't abundantly available to a crop, then photosynthesis and the resulting carbohydrate production is going to be impacted by that. In 90% of sap analysis samples that we see, roughly, of a standard kind of crop grown in average soil, iron and manganese, for instance, is deficient. And that the reasons for that, there are a lot of reasons for that, but those are two common micronutrients critical for photosynthesis that are often deficient. And so that's something to keep in mind as our goal is to optimize photosynthesis throughout the entire growing season. The more energy that we can produce, the better. The next piece is protein synthesis, and this is kind of the next function that crops are doing throughout the whole season. This is the process of taking that carbohydrate, the energy that they produce via photosynthesis, joining it with other mineral elements, calcium, nitrogen, other things, to build, eventually build proteins. And these are the processes by which plants actually build biomass. Like it says there, each reaction requires an enzyme and elemental cofactor. Magnesium, sulfur, molybdenum, and boron are kind of key pieces of the whole conversion of carbohydrates into proteins and also nitrogen into protein. Our goal in each 24-hour photo period is that all soluble nitrogen is converted to stable forms. We don't want any nitrogen circulate or free nitrate or ammonium circulating through the crop. There are implications there, strong implications for insect pressure and that sort of thing. This is not as much of an issue in soybeans, which is obviously a lower nitrogen input crop compared to something like corn, but I just mention that for reference. The next and sort of the last process I'll talk about is fruit, leaf, or grain quality. In this case, we're talking about grain quality. The protein levels in the finished grain, the test weight, whatever the metrics are for grain quality in your specific market. Some of the key nutrients there are calcium, potassium, manganese, zinc, and copper. I'm going to talk about this in more detail in a little while. Potassium particularly is going to be extremely important for the quality of that finished grain, how many sugars are moved into the pods during grain fill. So we've got these kind of three processes that plants are doing at various stages, or in some cases at every stage, every day throughout the time they're actively growing. But the timing of how we can affect some of these, affect the greatest crop response, we sort of divide into what we call critical points of influence, which are they often line up with kind of the phenological growth stages, but they're not exactly the same. These are the times that we can make an application and reduce the stress of a crop the greatest. And so we like to think about the genetic potential of a crop being 100%, kind of just like I said about the sunlight being 100% starting off. But we start with 100% genetic yield potential. And then every time that there's a stress on that plant, whether it's a nutrient deficiency, insect pressure, disease pressure, that 100% drops down. And we lose the opportunity for a healthy plant and for a higher yield. And so obviously stress equals yield loss there. And our goal is making applications and making nutritional recommendations at the right time to have the maximum impact on crop health. And we do that by thinking about these critical points of influence, which I'm going to run through. These are, again, these could be related or applied to other crops, but I'm going to try to keep it somewhat specific to soybeans. There are 6 or 7 of them. So some of this is very obvious, but it's a good reminder, I think. Germination and seedling development is obviously the first. Those are our goals. Getting strong root development, getting the root system colonized with a healthy microbiome. Biology is kind of the linchpin in making nutrients available, and it's something we talk about and focus on a lot. I probably won't be able to talk about biology as much as I'd like to, but it is something we focus on and work to foster and develop. But it's very important to us to inoculate our seed and get a strong biological presence there from the start to help really with disease resistance early on, resilience, drought tolerance, and also nutrient uptake. And then the other piece, one of our goal is to increase the stem diameter. The stem diameter really dictates how much the capacity of a plant to absorb nutrition from the soil. And so a larger stem diameter is going to correlate with that. Our critical nutrients at this stage are going to be, like I said, a microbial inoculant, but our photosynthetic elements that I mentioned earlier, and then calcium. Calcium is really important for strong germination. The next CPI is framing or vegetative growth, self-explanatory, but our goal really is rapid vegetative growth with tightly spaced internodes. The key takeaway and the key point here is there are two sort of plant hormones that drive vegetative growth, auxin and cytokinin. And an auxin-driven vegetative growth is going to be produced in a soybean, kind of a tall plant, not as bushy, not as many branches, one that's more prone to lodging, and the internodes are going to be really wide. And our goal is to not achieve that kind of vegetative growth, but to really try to use the other plant hormone, which is cytokinin. Cytokinin has a synergistic relationship with calcium. Auxin has a synergistic relationship with nitrogen. So we want to use calcium to push vegetative growth. Cytokinin is going to lead to a stockier plant, a bushier plant, one that has a lot more reproductive energy and is a lot less prone to lodging. Obviously, wide, thick, healthy leaves are a goal, and a rapid rate of biomass production, again, without added nitrogen. Critical nutrients, photosynthesis, again, calcium, boron, zinc, and nitrogen when, though not in the form of nitrates or ammonium. The next is bud initiation and development. And this is a very, very important one in soybeans because it's when we can probably have the greatest impact on the number of flowers, the pollen, well, not the pollination, but the number of flowers and the number of opportunities then for strong yield. And I'll circle back to this in a little bit, but our goals, of course, maximize the number of buds, the uniformity, the size, the energy, and maximize the blossom size for strong pollination. Our critical nutrients there are listed there. Again, photosynthesis, and then those others. Manganese is one that is especially important at this stage and at blossoming and pollination because it is the strongest, it has the strongest reproductive impact on a crop. It's the nutrient that will take a crop and shift it toward reproductive growth. And that really is our goal in addressing and thinking about critical points of influence for soybeans. We want to prevent too much vegetative growth, and we want to push it toward reproductive growth, especially when it's beginning to do that naturally during bud initiation and blossoming, which is the next. And obviously here we want to improve pollination, maximize the number of blossoms pollinated, and support the pollen and tube strength. Pollinators out there can honeybees or whatever kind of insects can tell the difference between a nutrient-dense, so to speak, flower and one that isn't. And if you ever watch pollination, a bee will choose a flower here but won't choose any of it next to it. And that's because it can tell the difference. They can sense that. And so our goal is to get the nutrition of the plant and the health of the plant such that every flower is attractive for pollination. so to achieve strong pollination. Critical nutrients there, again, manganese being very, very critical. The next is cell division. So this happens after the flower has been pollinated. And it's a period of time, a window of time. I'm not sure what it is exactly in soybeans, but normally it's 10 to 14 days in crops. It's when the single cell that was pollinated is rapidly dividing. And in that window of time, calcium is really, really, really important for maximizing the number of cells that ultimately determine the size and the health of the pod. So our goal, produce the largest quantity of cells during that window and maximize the high quality cells in pod development. The second to last is grain fill. Bud initiation and blooming and grain fill are the two most important points where we can have when we can have the greatest impact for soybeans. Grain fill is the time when the plant is moving sugars that are being produced as a result of photosynthesis in the leaves. They're moving those sugars, they're trucking them into the pods, and that's what's filling out and determining the quality of the finished grain. And so our goal is to, one, maximize photosynthesis, again, in the leaf, and then also maximize the sugar transportation into the grain. Often we see potassium being limited at this stage as a result of either low soil levels, poor soil microbiology, drought conditions, or any other stresses, but those are the big ones. So this is the time that we can really maximize grain quality metrics, protein content, test weight, all that sort of thing. Boron and potassium. Boron, so potassium is kind of like the freight train that sugars are loaded onto, and boron is kind of like the conductor. Boron sort of navigates the plant and tells the potassium with the sugars where to go. That's one way to think about it. Kind of the final CPI is grain finishing. Obviously, we want to produce grain with high protein content again, and this is probably going to sound kind of crazy. It did to me the first time I heard it, but a really healthy plant will produce finished grain while it's still green, and it won't die down. The grain will actually dry down while the plant is still alive and photosynthesizing. This isn't something we see very often, but a really healthy plant will do that. And so we're used to sort of seeing die down and dry down happening at the same time. So as kind of a lofty goal, this is kind of this is what we want to see. Impact the timing of senescence is kind of what I'm saying. And then sulfur and molybdenum there are bolded because those are two critical elements that determine protein content in grain. Kind of taking the nitrogen that's available and converting it into protein, along with, again, photosynthetic elements and the rest of those. So those are just all of the sort of nutrients that we look at, not as important there. Now, so we know what plants need at different times. We know what processes they're going through, what they need for photosynthesis, what they need for protein synthesis. How do we know then, and how do we make informed management decisions and recommendations based on that? We at AEA have kind of pioneered the use of SAP analysis here in the US. We've been using it for over 10 years now. I'm sure some of y'all have heard about it. It's increasing in popularity. More labs are doing it. But it is if you're not, I encourage you to, and I can talk to you more about that. But it's the most accurate in-season crop testing that we've got. Tissue testing is obviously kind of the standard, and it's what's been used for a long time. The difference between tissue testing and SAP analysis is this. For tissue testing, a leaf sample is taken, one leaf sample or a handful from one part of the plant, and sent to a lab where the leaf material is dehydrated, it's ashed, basically burned, and then that material is analyzed for its mineral content. And so what you end up getting as a result is all of the exterior of the leaf, everything on the interior of the leaf, everything inside the cells, everything outside the cells, all mixed together into a composite sample. And while this can be helpful, and you can sort of get some data from this over time, sap analysis is a lot more precise. For sap analysis, a young leaf sample on the plant and an old leaf sample is taken individually. Those samples are sent off to a lab in the Netherlands where the sap from literally the fluid that the plants are using to transport nutrients around itself is extracted, and then that is the material that's analyzed. So you don't get what's outside of the leaf, you don't get the construction of the leaf, you don't get the cellular construction, all of those elements that are part of that. It's like getting a blood test taken. It's the blood is, of course, our blood is the way that our body moves nutrients and other things around. Plant sap is the exact same thing. And so as a result, we can get a lot more information about nutrient uptake. It's very sensitive. Different nutrients have different types of mobility. Some can be moved from the old leaves to the young leaves. Some can't be moved from the old leaves to the young leaves. And we can see deficiencies or excesses 2 to 4 weeks before they manifest visually, which is really, really powerful because though there's no sort of silver bullet solution or anything in agriculture, this is as close as it gets to being able to sort of see into the future a bit. And we can see that deficiency, make an application, and prevent, again, that stress from affecting, the stress from that deficiency affecting the crop and the health of the crop and thus the yield. There are a lot of, we can take a SAP report and I can tell you, whether there's a good chance that you're going to have insect pressure, disease pressure. There are a lot of markers for these sorts of things. Very, very helpful tool. We have hundreds of thousands of data points over the last 10 years of using it, and we've been able to really move the needle on plant nutrition using it as a tool. And ultimately, all of this is really in service of input reduction. We can see if we have sort of optimal ranges for every single nutrient. So we can see if a potassium application early on, for instance, pre-plant, was really necessary. Was it excessive during the early stages of vegetative growth when the plant really didn't need it? And over time, we can kind of establish what's working and what's not working. So that really brings me to our approach to kind of nutritional management and what a sort of standard program that we like to adjust and tailor to the needs of specific growers we work with looks like. Primary goals, kind of to reiterate, we want to see strong cytokinin-driven vegetative growth, compact, bushy plants with a lot of heavy pod set, heavy bloom, and good pod fill. We want to support inherent resistance to insects and disease pressure without the use of insecticides or fungicides. We've been very successful doing this in a lot of different contexts, focusing on developing plants' natural immune systems, because plants, of course, have immune systems too, and we can increase their health and increase their nutrition to be resistant to fungal pathogens and insects. We want to maximize flowering and pod set and ensure complete pod fill. There are really 7 pieces that make up kind of a standard program for us. Number 1, an active inoculation of the soil with a biological soil primer. This is a mix of a few different products that is meant to support the establishment of biology. Both we're adding an inoculant in, but also supporting the native biology that's out there. The reduction in soluble fertilizer applications as a result of stimulating biology can be as much as 30% in 1 year as a result of really beginning to build biology. So biological soil primer, and this would be either field applied the fall before or the spring, onto the bare soil and lightly worked in, ideally, or it's put into a strip till type situation. Seed inoculation, I mentioned this a bit a little while ago. Every time, with soybeans, we recommend a high quality, nitrogen fixing organism inoculant. And a high quality inoculation of the seed is going to speed up germination, it's going to colonize that root system immediately, and it's going to improve nutrient uptake and seedling health. The third at planting would be a mineral planter application that does two things. Again, stimulates biology and supports biology and provides some early season nutrition, mostly those photosynthetic elements, phosphorus, micronutrients, that sort of thing. The fourth, if nitrogen or any soluble fertilizer is being used, like in the 2x2 or whatever, we want to stabilize those soluble elements so that they can be available to the plant for longer. We found that mixing soluble nitrogen or phosphorus with humic substances, carbohydrates, helps them remain in that root zone for a longer period of time, prevents leaching, and we can reduce the inputs, again, by about 30% as part of that. That's not as relevant, I realize, often in soybean production, but we do that sometimes. Now we get into kind of the importance of foliar applications, which are our preferred method of delivering those mineral nutrients to a crop when they need it. Foliar applications created with products that are very bioavailable, that are in the right form for plants to utilize them, sprayed on the leaf surface is the most efficient way to deliver precise amounts of nutrients to a crop in general. But in soybeans, we recommend 1 to 2 flowering foliar applications. So this is going to focus again on manganese primarily. It's important, though, that manganese is in the right form, in the reduced form, and not synthetically chelated. Synthetic chelation, which most manganese products are, EDTA chelated. The EDTA chelation is so strong that it won't let go of the manganese once the manganese is brought into the plant. And so it's important to have an organically chelated, amino acid chelated micronutrient there. So again, these are going to focus on reproductive nutrients, so manganese, calcium, seaweed, these sorts of things. Then getting into the pod fill, again, one application generally of potassium and manganese, among other things, is generally sufficient in most contexts to encourage that strong sugar movement into the pods. And then threaded through this whole program would be 2 to 3 sap samples, again, to monitor the health of the crop. And ideally, we time these before the foliar application so we can adjust based on the results. And this gives us just as clear of a picture of what's really going on inside the plant as you can get. And we can add or remove things as needed. Again, sort of realistic expectations here. Better soil tilth and reduction of compaction as a result of adding biology and stimulating biology. It's really amazing what we've seen in just a year, one season on that metric there. Also improved residue decomposition from that. And then strong, consistent germination. Like I said, reduction of fertilizer, pod count increase. Generally, we see 20 to 30% pod count increase as a result of focusing on the nutrition of crops, of soybeans. Again, compact plants, not prone to lodging, and best case scenario, anywhere from 15 to 20% yield increase year one. So, did you have your hand raised? Oh, sorry. And then the big thing, a big also sort of side effect of this is beginning to get a deeper understanding of your crop's nutritional requirements specific to your farm. And this is really powerful because, as all of you know, one field to the next can vary night and day. And so beginning to dial in the understanding of your plants, your crops, in your context, your soil environments is really, really valuable. So to sort of just end, this is just a picture of a pretty healthy trifoliate from a grower here in North Carolina I worked with. Big leaves, you can kind of see the glossy sheen on those leaves. That's really what we're going for. That's symptomatic of a plant being able to have a high lipid content, which means that it's got plenty of energy, it's efficiently photosynthesizing. This was kind of a partial program that he was doing and had pretty good success with. And then the last picture this is just very illustrative of the difference between auxin and cytokinin-driven vegetative growth. This was a trial that we did here in North Carolina. And on the left, you've got a plant in a control field, and on the right, you've got the AEA treated, and you can just tell the height difference, and the internodes are so much farther apart in the control. The pod counts differed in the right in the AEA treatment. We had 111 pods in this picture. In the control, we had 89, and so significant difference. The yield ended up being, for the most part, the same with in the better sections of the AEA treated field, we saw about a 15% yield increase. But the point really here is the height difference. And this is just from a very similar kind of program that I just recommended or just kind of went through. And I was really shocked to kind of see this. It surprised me. But a stocky, cytokinin-driven, calcium-driven vegetative plant is really what we're going for. So I know I kind of ran through a lot of that really quickly, but happy to answer any questions, talk to any of y'all individually if you have any. Yes, sir.
If you have a soybean crop that's tremendously large, what does that cost?
What does it cost as far as
in terms of the
yield? Well, what's happening there is, a section of the roots are getting dislodged from the soil. And so the ability for those roots to take up nutrients and to interface with that soil is going to be impacted by that. And that's a very stressful event physically for a plant. So I would imagine the yield implications there are pretty significant. Yeah. Yes, sir.
I noticed on all your scenarios from basically start to finish, your essential nutrients, you had boron in every one of them.
Yep.
We typically spray boron on cotton and peanuts. What is boron doing for soybeans?
Sure. Boron, you have to be careful with boron in soybeans and beans in general. There's a certain, they have a lower tolerance for boron toxicity. The reason it's in every one of those is, well, there are a couple reasons. The first is boron's a negatively charged element. And so it's one of the ones that's going to leach every year. And so we find that boron uptake generally is very poor, number one, because it's not as available in the soil. And two, it is, lost my train of thought. But that's the big thing. And so in soybeans, boron plays a lot of different roles in all crops, but one of the biggest things is it can improve calcium uptake. And calcium becomes one of the biggest limiting factors really in so many agricultural scenarios that I've experienced as a consultant, because the strong uptake of calcium, which leads to all of these different things, the compact vegetative growth, the strong cell division, strong pod set, all these different very, very important things, is calcium uptake is predicated, it's dependent on strong soil biology. And we don't see the robust biology that we'd like to in most soils. And so it's also very important for protein synthesis and nitrogen synthesis. So, yeah. good question there. Anything else?
In a lot of your slides, you showed seaweed.
Yeah, good question. Seaweed is a big component of a lot of our foliar applications and treatments because for whatever reason, and this isn't totally understood, seaweed will stimulate the immune system of a plant. It will, it just makes plants healthier. We don't fully understand why sea products in general are so good for crops. We use 3 different sea products. One is like a ground up fish hydrolysate type product, fish emulsion, and then seaweed, and then ocean water concentrate. But it will stimulate the immune system of plants, and it also plays a huge role in the whole cytokine to auxin ratio that I mentioned about the vegetative growth, and it encourages a plant to switch over to reproductive growth. And so for soybeans specifically, that's our goal in our climate where we really will get this kind of rank vegetative growth is to really push the reproductive energy in the crop.
How does the seaweed come, and can you replace it with algae weed?
Can't be replaced. It comes in a tote or a barrel, however much you want. I'll have it sent right to your farm. Thank you all very much. Appreciate it.
All right, let's give a round of applause for all our speakers.